Hello friends, NCERT-Botany, next episode, second episode in plant growth and development. One more episode needed to finish this chapter. Then we'll be entering into genetics, first chapter, principles of inheritance and variation. So what all things we'll be learning in this episode? For that, I will share the screen for convenience. I think screen came. So what all things we'll be learning under plant growth and development, second episode. 7 ideas has been given. And last is auxin, just an introduction. Next episode, we'll be discussing all these 5 plant hormones. So we can start with the first item, that is differentiation, meaning, purpose, aim, how, all these things. So first, meaning of the term differentiation. So differentiation means it is an act, it is an event, it is a procedure leading to maturation. More expanded form, it means cells derived from meristems will undergo permanent changes in many aspects by which respective cell is able to do specific function allotted for it. Typical example, development of tracheal elements connected with the xylem. The cell has to lose the protoplasm. Along with it, development of elastic, lignocellulosic secondary cell wall. Purpose is to provide a strong wall to face even extreme tension while doing conduction of water for long distance. Another example is development of parenchyma with the chloroplast, parenchyma cells for photosynthesis. So I think differentiation, meaning, purpose, all are clear to you. Now, how these cells get modified? See, cells modified in connection with the function it has to do, and even considering the position. Now we have to proceed to next aspect, types of differentiation. It is undifferentiation, differentiation already over, dedifferentiation and redifferentiation. So 3 more terms we have to discuss. One by one we will be proceeding. So I think first page is clear to all of you. Now we can proceed to second page, undifferentiation. Undifferentiation literally means not differentiated. So what is the biological meaning? Cells which, number 1, do not have specialized function. Number 2, no specialized structure. Number 3, not mature. So indirectly not able to do any function. Meristem, which results or producing cells which undergoes differentiation to become functional. How cells get differentiated from an undifferentiated area? That aspect still not clear. Some speculations are there. There it mentioned genes may be involved. That is regarding the term undifferentiation. Now comes the third term, dedifferentiation. So whenever the term DE, normally the term will be added as a prefix, gives an opposite meaning. And DE, D according to different situations, meaning may change. And typical example, sometimes the meaning is not, sometimes the meaning is minus, sometimes the meaning is inferior, sometimes opposite. Typical example, RNA and DNA, deoxyribonucleic acid. So here it means living differentiated cells which lost the power to divide will regain the power to divide under certain situations. Typical examples, formation of intervesicular cambium and core cambium from fully differentiated parenchyma cell. I think de-differentiation also clear. Now comes the last term in this series, that is redifferentiation. Product of differentiated cells will lose the power to divide again, called as redifferentiated. So product of de-differentiated cells will lose the power to divide again, and it is called redifferentiated. Typical example is vascular cambium into secondary xylem and secondary phloem that will not divide. That is regarding 4 terms: undifferentiated, differentiated, de-differentiated, and redifferentiated. Exam point of view, whether theory and runs, it is important. 3 specific area it comes in NCERT. One is cell as the basic unit of life. One is anatomy part. Third is plant growth and development. Now comes the term development. So what is development? Development includes all the changes that an organism passes through during its entire life cycle for plants from seed to senescence. And there is an NCERT chart given there. Most of the time familiar to us, but one term that is exception, plasmatic growth. So what is the term plasmatic growth means, or what is the meaning? Plasmatic growth is the process of increase in the size of the cell which leads to differentiation. In plants, plasmatic growth refers to increase in cell division of meristems leading to differentiation. Then it continues, then aging, senescence, that way it proceeds. So I think plasmatic growth is clear. The term not directly explained in NCERT because that term is there in the diagram, plasmatic growth. Next is development stages of plants. So a flow chart, seed germination, seedling, juvenile phase leading to maturation. Maturation produces flowers, flowers develop seeds, seeds then gradually that plant undergo aging and senescence. This type of sequence we can find in many places, including an individual cell. Next is plasticity. So what is plasticity? What is the need? How, etc.? It is a theory entrance question, direct NCERT. Idea is simple. An organism to grow and develop needs adaptation and adaptability with the environment. This ability is called plasticity. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called plasticity. Most basically, it means adaptation, adaptability to the situation connected with the environment. Survival, that is the basic meaning. And to show plasticity, NCERT is shown 2 sets of diagrams. First pair, second pair. First pair, it is labeled as juvenile and adult. Second item, they mark it as terrestrial habitat and aquatic or water habitat. And what it means. Plants like cotton, coriander, larkspur. This change the shape of leaves takes place when the plant from juvenile to adult. Shape of the leaf changes. While in a plant called buttercup, otherwise called ranunculus, it is an aquatic plant where 2 types of leaves. Leaves on the surface will be entire, or that is above the water surface will be entire, just on the surface, etc. But leaves underwater, that will be splitted into hairs. Why that splitting like hairs? Due to heavy water currents. Otherwise, suppose if it is entire leaf, it will be teared off. So to prevent it, underwater leaves will be segmented like hairs. So these are the 2 types. One is due to growth pattern, Other is due to environmental factors. And presence of two types of leaves within same plant, it is called heterophyly. Thus, what is development? Development means totality of growth and differentiation connected with intrinsic and extrinsic factors. Intrinsic basically means internal, and that can be two types. One is within cell, that is called intracellular, otherwise genetical, intra. Second is intercellular, that is between cells. Example is PGR, plant growth regulators or chemicals. So that is regarding intrinsic. Extrinsic means totally outside the plant, connected with nature like environment, temperature, all this. I think that page is clear. Now we can proceed to next page. So it is represented as a chart. So very easy to identify, to understand. Now we have to proceed to plant growth regulators, last part of this episode, PGR. So first we should identify what is a regulator. Regulator is familiar to us. If we have a ceiling fan, there is a knob to regulate the speed. Secondly, we will be studying operon concept, gene regulation. So this plant growth regulator PGR, also known as phytohormones, plant hormones, all are synonyms, PGR. And what are the features of PGR? Theory question, entrance question. PGR are usually small, simple, organic in nature, with a different chemical composition required in low concentration. Plant growth regulator, otherwise hormone, they are very closely linked with metabolism. Metabolism means physiology. Physiology means it explains or try to explain how life is maintained. PGR may be produced at one site and may be utilized in another site. Different types of chemicals which will act as plant hormones. Thus, these plant hormones has got different properties. That detailed study will be in next episode. And what are the common plant hormones? For us, 5 plant hormones are there: auxin, gibberellic acid, cytokinin, ethylene, abscisic acid. Auxin is indole derivative, gibberellic acid is terpenes, cytokinin adenine derivative, ethylene is gaseous, abscisic acid is derivative of carotenoids. Among these 5 plant growth regulator, 5 plant hormones, cytokinin is basic in nature, exception. Ethylene is gaseous in nature, exception. Both are exception. I think this page is clear. Now we can proceed to next page, classification of PGR. Based on the physiological action, PGR can be classified into 2. One is promoters, other is inhibitors. Promoters means they will enhance, they will allow something to take place. Inhibitors means stop, they will not allow. But remember, these two are the sides of a coin. Likewise, it is for the beneficial nature of the plant. And what are the famous promoters? Auxin, gibberellic acid, cytokinin. Inhibitors, ethylene, abscisic acid. But again, here one exception for ethylene. Ethylene can act as a promoter as well as an inhibitor. I think that part is clear. Now, the first hormone, just an introduction, auxin. It came from a Greek word, meaning, or the term is auxin, means to grow. Last episode, we studied an instrument called oxinometer to measure growth. And after this, 5 theory question, 5 entrance question with the key. So these are the items you have to learn in this episode. So we'll just pass through plant growth development, second episode, 7 aspects, starting with the differentiation, meaning, why it is an important event, what is the purpose. Then next is type of differentiation, undifferentiated, dedifferentiated, redifferentiated. Now what is undifferentiated? What are the features? And next is dedifferentiation. What happens in dedifferentiation? That is, some of the cells which lost the power to divide will regain the power to divide under certain situation with example. Then again, redifferentiation, again losing the power to divide. Then we pass through an area called development. What is development? Then a flowchart, where a term we specifically discussed that is called plasmatic growth. Then developmental stages of a plant, again a chart. Then comes the famous term plasticity. What is plasticity? Adaptation, adaptability with the two sets of leaves. One is growth pattern, one is environmental pattern, heterophyly. Then last part, intrinsic as well as extrinsic. Intrinsic is both intracellular and intercellular. Extrinsic means climatic condition. Then plant growth regulators. What are plant growth regulators? What is their other name? Phytohormone, plant hormone, PGR. What are the features of PGR? And where it works, where it produced. And common PGR. Chemically, they are different. Properties also different. Oxygen, gibberellic acid, cytokinin, ethylene abscisic acid. And what is the exception of cytokinin? That is basic. What is the exception of ethylene? It is gaseous. Then comes classification, promoters and inhibitors. Oxygen, gibberellic acid, cytokinin promoters, ethylene abscisic acid, normal inhibitor. But again, here we can find exception ethylene. Ethylene can work in both ways. Then we introduce the term auxin, Greek word, and what it means. Oxinometer instrument also we discuss. Then 5 theory question, 5 entrance question. So if you like this episode, subscribe all, like it, share it, comment on it. And we will be continuing with plant growth and development, 3rd episode, hormones, last. So we'll be meeting again. Thank you.